As we further elucidate the varied genetic landscape of heritable cardiac disease, mutations in alpha-actinin 2 have been identified in association with arrhythmia, cardiomyopathy, and heart failure. Alpha-actinin 2, encoded by the ACTN2 gene, is a sarcomeric protein that cross-links actin and titin filaments in the Z-disc. Mutations in ACTN2 have been associated with dilated and hypertrophic cardiomyopathy, but how disease develops is still emerging. This manuscript by Lindholm et al. From Stanford University seeks to explore this. These researchers queried the Stanford Center for Inherited Cardiovascular Disease database for rare or novel ACTN2 variants and identified two families. One family with an individual homozygous for a novel glutamine 860 stop variant, and the other family with individuals heterozygous for a 4.3 kilobase deletion spanning exons 8 through 10. Lindholm and colleagues then generated patient-derived induced pluripotent stem cells that were differentiated into cardiomyocytes. Lindholm et al. Found that both variants were associated with cardiomyocyte hypertrophy and decreased contractility. The exon 8 through 10 deletion ACTN2 variant demonstrated sarcomeric disarray with larger Z discs and thicker I bands. The alpha-actinin 2 interactome was explored, and in wild-type cardiomyocytes, alpha-actinin 2 was found to interact and co-localize with alpha-actinin 1 and connexin 43. However, in the glutamine 860 stop variant, this interaction was lost, and there was a compensatory increase in the alpha-actinin 1 expression. Overall, these data suggest a role for alpha-actinin 2 variants in sarcolemmic dysregulation. These findings shed important light on how disease-associated variants in ACTN2 can cause arrhythmia and cardiomyopathy. Altogether, this study supports inclusion of ACTN2 as a putative cardiomyopathy-causing gene.